Hello, welcome to another ANP2 Google Hangout session. My name is Colin Cairns. I'm a tutor at Baker College of Auburn Hills Learning Center. Today we're going to cover Chapter 21 and 22. Chapter 21 is Water, Electrolyte, and Acid-Base Balance. And Chapter 22 is the Reproductive System. I'm about to pull down a PowerPoint to share with you. Introduction to Chapter 21. The term balance suggests a state of equilibrium for water and electrolytes. That means equal amounts enter and leave the body. There are mechanisms that replace the lost water and electrolytes and excrete excesses to maintain this balance. But this results in stability of the body at all times. And you have to keep in mind that water and electrolyte balance are interdependent. If you are lacking water, it will affect your electrolytes. And if you are lacking in certain electrolytes, it'll affect the amount of water that your body retains. Body fluids are not uniformly distributed. They occupy compartments of different volumes that contain varying compositions of electrolytes. Water and electrolyte movement between these compartments is regulated. That means that their distribution is stabilized and the composition of body fluids is maintained. So we have 40 liters of water in an average adult body. 2/3 is intracellular fluid inside the cells, and 1/3 of that is extracellular fluid. It occupies the interstitial spaces. An average adult female is about 52% water by weight, while the male actually has more water by weight, 63%. And here's just another slide showing how that you have 63% of your body water in intercellular fluid inside the membrane of your cells. And then that rest of the 37% is divided between interstitial fluid, plasma, some lymph, and some transcellular fluid. And here's a different bar graph showing the ratio between the extracellular fluid and intercellular fluid and the electrolyte composition of both of those. So for example, sodium has a ratio of 14:1 between extracellular and intercellular fluid, whereas potassium has the opposite ratio, a double inverse of that ratio, 28:1. Most of your potassium is in the cells, very little is outside the cells. Another one that is great ratio is the magnesium. It's a 1:19. Very little outside the cells. Most of your magnesium is in your cells. So extracellular fluids are generally similar in composition. There's a high concentration of sodium, calcium chloride, and bicarbonate, where your intercellular fluids have high concentrations of potassium, magnesium, phosphate, and sulfate. This next slide shows the movement of fluid between compartments. And there's two major factors that regulate the movement of water and electrolytes. That would be the hydrostatic pressure, the pressure that's exerted within either the interstitial spaces or the outward force against the walls of the capillaries. And then there's the osmotic pressure. That's the pressure that water, that forces water in and out of cells. Water balance exists when the water intake equals the water output, and homeostasis requires control of both intake and output. The volume of water gained each day varies among individuals, usually about 2,500 milliliters daily for an adult. So if you think of 2,500 milliliters is actually 2.5 liters, or the same as 5 bottles of drinking water, basically. So 60% of your water comes from drinking, 30% from moist foods, and 10% as a byproduct of oxidative metabolism of nutrients. And when you're taking in 2,500 milliliters daily, you should be excreting 2,500 milliliters daily. 6% is lost in sweat, 6% is lost in feces. Water is lost through your skin and lungs, about 28%, and urine accounts for 60% of the water loss. So the regulation of water intake, your body should lose as little as 1% of its water. There's an increase in the osmotic pressure of extracellular fluid due to water loss, which stimulates osmoreceptors in your thirst center, so it makes you want to drink more water. And activity in the hypothalamus causes the person to feel thirsty. Drinking and the resulting distension of the stomach by water stimulates nerve impulses that inhibit the thirst center. And then water is absorbed through the walls of the stomach and small intestine, and then your osmotic pressure of the extracellular fluid returns to normal. Again, we talked about how water is lost. This slide just shows the numbers again. And then here's the events in the regulation of water output and some of the factors that cause dehydration. The extracellular fluid becomes osmotically more concentrated. The osmoreceptors in the hypothalamus are stimulated by the increase in the osmotic pressure of the body fluids, which signals the posterior pituitary gland to release the antidiuretic hormone into the blood, which is carried to the kidneys. That ADH causes the distal convoluted tubules and collecting ducts to increase the water reabsorption, which increases urine output and further, which decreases urine output, sorry, because you're reabsorbing water and less is excreted. And then, so that minimizes further water loss and prevents dehydration. When you have excess water intake, the extracellular fluid becomes less concentrated. That change stimulates the osmoreceptors in the hypothalamus, which tells the posterior pituitary gland to decrease the release of ADH, and your renal tubules then decrease water reabsorption, and your urine output increases, and the excess water is excreted. So this slide shows a little bit about electrolyte balance. The balance exists when quantities of electrolytes the body gains equals those lost. Again, foods, fluids, and metabolic reactions contribute to electrolyte intake, and the output through perspiration, feces, and urine, and the body tries to maintain a balance. So let's talk about some of the important electrolytes to cellular function. Sodium, potassium, calcium, magnesium, chloride, sulfate, phosphate, bicarbonate, and hydrogen And these ions are primarily obtained from foods, but some are also in water and other beverages. Some of the bottled water today is enhanced. You'll read the label. It usually says that it's enhanced with minerals, and a lot of times it'll be enhanced with magnesium. Ordinarily, a person obtains sufficient electrolytes by response to hunger and thirst. Severe electrolyte deficiency may cause you to crave salt. The body will lose electrolytes by perspiring, typically on a warmer day and during strenuous exercise, and some are lost in feces. But the greatest output is a result of kidney function and urine output. So let's talk about a little bit of the potassium, sodium, potassium, and calcium intake and regulation. These ions are all vital for nerve impulse conduction, muscle fiber contraction, and maintenance of cell membrane permeability. So if you have a potassium ion concentration increase, your adrenal cortex is signaled, aldosterone is secreted, the renal tubules increase reabsorption of sodium ions, and then increase secretion of potassium. So the sodium's conserved and potassium's excreted. If calcium ion concentration decreases, then the parathyroid glands are stimulated, parathyroid hormone is secreted, activity of bone reabsorbing osteoclasts increases, and then that calcium's used up so that your concentration returns to normal. Or the intestinal absorption of calcium increases, and then the calcium returns to normal. Or renal tubules conserve calcium, increase secretion of phosphate. That increased phosphate is excreted in urine, and normal phosphate concentration is maintained. There's an addition, after the activity of bone reabsorbing osteoclasts increases, there will be addition of phosphate into the bloodstream, and that helps maintain normal phosphate concentration. Let's talk about the acid-base balance. Electrolytes that ionize in water and release hydrogen ions are acids. Substances that combine with hydrogen ions are bases. The acid-base balance entails regulation of the hydrogen ion concentration of body fluids. High hydrogen concentration is also your pH level. This is important because slight changes in hydrogen ion concentrations can alter the rates of enzyme-controlled metabolic reactions, shift the distribution of other ions, or modify hormone actions. So here's some of the sources of hydrogen ions. The aerobic respiration of glucose results in carbonic acid and contributes to the hydrogen ion concentration in the internal environment. Same with the anaerobic respiration of glucose, which contributes to lactic acid. The incomplete oxidation of fatty acids, you have acidic ketone bodies, oxidation of sulfur-containing amino acids, you get sulfuric acid. The hydrolysis of phosphoproteins and nucleic acids, you'll have phosphoric acid, all contributing to the hydrogen ion concentration in the internal environment. The strengths of acids and bases. Strong acids ionize more completely and release more hydrogen ion, while weak acids ionize less completely and release fewer hydrogen ions. Strong bases ionize more completely and release hydroxide, and the weak bases ionize less completely and release fewer hydrogen oxide. So if we have a high concentration of hydrogen ions, if we look back at the chapter on pH levels, if we have highly acidic, and if we have low concentration, we have more base. The regulation of hydrogen ion concentration, either an acid shift or an alkaline shift in the body fluids, could threaten the internal environment. Normal metabolic reactions generally produce more acid and base. The reactions include cellular metabolism of glucose, fatty acids, and amino acids. The maintenance of the acid-base balance usually eliminates acids in one of three ways. The acid-base buffer system, there's the respiratory excretion of carbon dioxide and the renal excretion of hydrogen ions. And let's look at these acid-base buffer systems. There's a bicarbonate buffer system. The bicarbonate ion converts a strong acid to a weak acid. Carbonic acid converts a strong base to a weak base. Hydrogen ion and the HCO3. That's a carbonic acid. They form the weak acid, carbonic acid. And the phosphate buffer system, the monohydrogen phosphate ion converts a strong acid to a weak acid. So you have the hydrogen ion combining with the dihydrogen phosphate. Or I'm sorry, that's the monohydrogen phosphate converts to dihydrogen phosphate. And in your protein buffer system, and then the NH3 group releases a hydrogen ion in the presence of excess base, while the COO group, carboxyl group, accepts a hydrogen ion in the presence of excess acid. And here's a chart of that. Bicarbonate ion converts a strong acid to weak base. Carbonic acid converts a strong base to weak base. Monohydrogen phosphate ion converts a strong acid into a weak acid. Dihydrogen phosphate converts a strong base into a weak base. The NH3 group of amino acid or protein releases a hydrogen ion in the presence of an excess base, while the COO group of an amino acid or protein accepts a hydrogen ion in the presence of excess acid. Let's talk about respiratory secretion of carbon dioxide. The respiratory center in the brainstem helps regulate hydrogen ion concentrations in the body fluids by controlling the rate and depth of breathing. If body cells increase the production of carbon dioxide, as occurs during periods of physical exertion, carbonic acid production increases. As the carbonic acid dissociates, the concentration of hydrogen ions increases and the pH of the internal environment drops. Such a concentration of carbon dioxide in the central nervous system and the subsequent increase in hydrogen ion concentration in the cerebral spinal fluid stimulate chemosensitive areas in the respiratory center. In response to stimulation, the respiratory center increases the depth and rate of breathing so that lungs excrete more carbon dioxide. You give off more. And then hydrogen ion concentrations in body fluid will return toward normal because the released carbon dioxide is in equilibrium with the carbonic acid and the CO2. And this chart just shows that what I just said, basically. Cells increase production of CO2, and the CO2 reacts with water to produce the H2CO3, and then the HC2O3 releases hydrogen ions, respiratory center stimulated, rate and depth of breathing increase, and then more CO2 is eliminated. Renal excretion of hydrogen ions. The nephrons help regulate the hydrogen ion concentration of body fluids by excreting hydrogen ions in the urine. If you have a high intake of proteins, there will be an increased metabolism of amino acids. This causes an increased formation of sulfuric acid and phosphoric acid. Then you have an increased concentration of hydrogen ions in the body fluids. That in turn increases secretion of hydrogen ions into the fluid of the renal tubules. And then you have increased concentration of hydrogen ions in the urine. And then the concentration of hydrogen ions in body fluid returns to normal. There's various regulators of hydrogen ion concentration that operate at different rates. The acid base functions rapidly. The respiratory and renal mechanisms function more slowly. The first line of defense against the pH shift is the chemical buffer system. And the second line of defense is your physiological buffers, either the respiratory mechanism or the renal mechanism. And it talks about a little bit of the acid-base balance. So you have high pH, more acidic, low pH, more alkaline. Wait, did I say? Okay. Higher ions, lower pH, more acidic. Lower ions, higher pH, more base. So it's an inverse between the hydrogen ion concentration and pH level. And you can survive between 6.8 and 8. A normal pH is between 7.35 and 7.45. Acidosis results from the accumulation of acids or loss of bases, both of which cause abnormal increases in the hydrogen ion concentration of body fluids. So the more acidic, more hydrogen ions, lower pH, your pH drops, it's acidosis. Alkalosis, on the other hand, results from a loss of acids or an accumulation of bases, accompanied by a decrease in hydrogen ion concentrations. So you have accumulation of base, loss of acids, decreased concentration of hydrogen ions, pH rises, and that's alkalosis. There's two major types of acidosis. One's respiratory and one's metabolic. So what happens, you have a decreased rate of depth of breathing or obstruction of air passages or decreased gas exchange. You have an accumulation of CO2. That results in respiratory acidosis. Metabolically, if your kidney fails to excrete the acids, or there's excessive production of acidic ketones, as in diabetes, you have an accumulation of non-respiratory acids, leads to metabolic acidosis. There's also prolonged diarrhea with loss of alkaline intestinal secretions, or prolonged vomiting with loss of intestinal secretions will raise the level of acid in your system, and that can lead to an excess of loss of bases and metabolic acidosis. The balance is upset in favor of too much acid. low pH, high hydrogen ion concentration. Alkalosis, it's the opposite of, not breathing enough. Now you're breathing too much. It can be a result of hyperventilation. So there's a great loss of hydrogen ions or a gain in bases, both accompanied by a rise in the pH of blood. So say there's anxiety, fever, poisoning, or high altitude results in hyperventilation, excessive loss of CO2. There's a decrease in the concentration of bicarbonate and decrease in concentration of the hydrogen ion concentration. So now you have a high pH and respiratory alkalosis. Another way this can happen is either gastric drainage or vomiting with loss of gastric secretions. So you lose acids. So there's net increase in alkaline substances and results in metabolic alkalosis. Okay, so real quick, we're going to talk about Chapter 21's outcomes to be assessed. Introduction, explain the balance between acids and bases, explain the importance of water and electrolyte balance, distribution of body fluids, how body fluids are distributed in compartments, intercellular, extracellular, and transcellular. Explain how fluid composition varies among compartments and how fluids move from one compartment to another by osmosis or hydrostatic pressure. Water balance, list of routes by which water enters and leaves the body. Explain the regulation of water input and water output. List the routes by which electrolytes enter and leave the body. Explain the regulation of the input and output of electrolytes. Explain the acid-base balance. Identify how pH number describes the acidity and alkalinity of a body fluid. List the major sources of hydrogen ions in the body. Distinguish between strong acids and weak acids. Explain how chemical buffer systems, the respiratory systems, and the kidneys keep the pH of body fluids relatively constant. And describe the causes and consequences of an increase or decrease in body fluid. Well, that's all for Chapter 21. So I'm going to exit this one, and I'm going to start on Chapter 22. Okay, Chapter 22, the reproductive system. The male and female reproductive organs and systems are connected by a series of organs and glands that produce and nurture sex cells and transport them to the sites of fertilization. The male sex cells are the sperm, and female sex cells are eggs or oocytes, which is Latin for egg cells. Sex cells have one set of genetic instructions carried on 23 chromosomes compared to 2 sets of 46 chromosomes in other body cells. When sex cells join in fertilization, the amount of genetic information held in 46 chromosomes is restored. And the sperm and oocytes are produced by a special type of cell division called meiosis. Meiosis includes 2 successive divisions called the first and second meiotic divisions. The first meiotic division separates homologous chromosome pairs. So these pairs are the same gene for gene. They might not be identical, however, because a gene may have variants, and the chromosome that comes from the person's mother may carry a different variant from the corresponding gene from the father's homologous chromosome. So before the first part of meiosis, or meiosis 1, each homologous chromosome is replicated, so it consists of 2 DNA strands called the chromatids. Each chromatid has the complete genetic information associated with that chromosome. The chromatids of a replicated chromosome attach at regions called centromeres. And here you can see chromosome, each with 2 chromatids. Here's your paired homologous chromosomes. And after the split, you have 23 chromosomes here, and then 23 chromosomes here, each with 2 chromatids. And then in your second meiotic division, and now you're going to have 23 and 23, 2 and 2 for each one, and each chromatid now has an independent chromosome. So meiosis 1 separates homologous, the same gene for gene pairs. There are 4 phases in its division, and prophase 1, metaphase 1, anaphase 1, and telophase 1. One way to remember this is a mnemonic: pass my anatomy test. Prophase 1. Let's do the animation first.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase 1 of meiosis 1 begins with the condensation of chromosomes and the vesiculation of the nuclear membrane. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase 1, the spindle apparatus continues to form, and bivalents become attached to kinetochore microtubules. One pair of sister chromatids is connected via kinetochore microtubules to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase 1. The chromosomes align at the metaphase plate during metaphase 1. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in 2 cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis 2.
And here's a shot of crossing over, mixing up genetic traits. In A, you have homologous chromosome pair. B, you're showing the chromatids crossing over. And what happens when they cross over? There's a recombination of genes. The different colors indicate that one of the homologous chromosomes comes from the individual's father and one from the mother, but I'm not sure which is which. But that's what you're showing here. Okay, let's talk about the second meiotic division. Meiosis 2 begins after telophase 1. The division is similar to mitosis. Again, there are 4 phases in this division. Again, prophase, metaphase, anaphase, and telophase. This division completes with each sex cell having one set of genetic instructions, or 23 chromosomes, compared to 2 sets in other cells. And here's an animation on meiosis 2.
At prophase 2, the chromosomes condense again, and the nuclear membrane vesiculates. During prometaphase 2, the spindle apparatus forms, and the sister chromatids become attached to kinetochore microtubules. In this case, a pair of sister chromatids is attached to both poles, not just one pole as in prometaphase 1. At metaphase 2, the sister chromatids are aligned along the metaphase plate, with each chromatid being attached to one pole. The sister chromatids separate or disjoin and begin moving to the poles of the cells during anaphase 2. During telophase 2, the nuclear membrane reforms and chromosomes decondense. In animal cells, a cleavage furrow is formed that causes cytokinesis or cellular division. As cytokinesis is completed, meiosis 2 results in the formation of 4 haploid cells. Thus, meiosis begins with a diploid mother cell that has replicated its chromosomes and ends with 4 haploid cells containing one set of chromosomes each.
Okay, so here's a slide showing a parent cell, the paternal chromatids, the gene for the blood type, the gene for the eye color, the gene for the hair color, and the maternal chromatids, again, the blood type, eye color, and hair color. And here's a result of crossing over. So you have a maternal gene for blood type, and a paternal gene for blood type. And then when they split, as you can see, the split leaves a new gene with parts of both. A new chromatid, I mean, with parts of both paternal and maternal genes. And let's look at how meiosis works.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber, and in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Okay then. I'm not going to play this one. Again, it's the different stages of meiosis. Okay, chapter 22.2. I talk slide about the organs of the male reproductive system. You have the ductus, the vas deferens. That's important. The function of the two ducts that are formed. They're an ejaculatory duct. They pass through the prostate gland and enter the urethra through a slit-like opening. And then let's look at the seminal vesicles right here. They're sac-like structures about 5 centimeters long, each attached to a ductus deferens near the base of the urinary bladder. The glandular tissue lining the inner wall of the vesicle secretes a slightly alkaline fluid, and this fluid helps regulate the pH of the tubular contents as the sperm travels to the outside. And additionally, the seminal fluid neutralizes the acidic secretions of the vagina to help sustain sperm cells that enter the female reproductive tract. There's also some fructose, monosaccharide, and prostaglandins, which stimulate muscular contraction of the female reproductive organs, aiding the movement of sperm cells toward the egg cells. And here's a look at the testes. And there's the ductus vas deferens. And okay, and descent of the testes. And the structure of the testes. The epididymis. It plays a role in the maturing of sperm cells. Sperm cells, once they age, can move independently and fertilize egg cells. And here's the formation of sperm cells from a primary spermatocyte, first meiotic division. You have 23 chromosomes, each with 2 chromatids. Then that separates again, the second meiotic division, and then you have 4, each with 23 chromosomes and an independent chromosome. And let's talk a little bit about spermatogenesis.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
And here's the structure of a sperm cell, the nucleus and the flagellum. And you have the acrosome, the midpiece, and the tail. And here's the male internal accessory organs, including the epididymis, the ductus deferentia, the seminal vesicles, the prostate gland, and the bulbourethral glands. And here's epididymis. They're tightly coiled tubes. They connect to the ducts within the testes. They promote the maturation of the sperm cells. And the ductus deferentia, they extend from the epididymis to the ejaculatory duct. And seminal vesicles, they're attached to vas deferens near the base of the urinary bladder. They secrete alkaline fluid. They secrete fructose and prostaglandins. And the contents empty into the ejaculatory duct. Again, we talked about that alkaline fluid neutralizing the acid of the vagina. Prostate gland surrounds the proximal portion of the urethra, secretes a thin, milky alkaline fluid. Secretion enhances fluid mobility, composed of tubular glands in the connective tissue, and also contains smooth muscle. This acidic wait a minute. Okay. It's actually slightly acidic. Not alkaline. It contains citrate, a nutrient for the sperm, and a prostate-specific antigen, which PSA, which is an enzyme which helps liquefy semen following ejaculate, breaks down the semen following ejaculation. The bulbourethral glands are 2 small structures, each about 1 centimeter in diameter. They are inferior to the prostate gland, lateral to the membranous urethra, and enclosed by muscle fibers of the urogenital diaphragm. They're composed of many tubes whose epithelial linings secrete a mucus-like fluid. This fluid is released in response to sexual stimulation and lubricates the end of the penis in preparation for sexual intercourse. However, females secrete most of the lubricating fluid for intercourse. Semen, That's the fluid that the urethra conveys to the outside during ejaculation. It consists of sperm cells, secretions of the seminal vesicles, prostate gland, and bulbourethral glands. It's slightly alkaline, contains prostaglandins, contains nutrients. Volume is 2 to 5 milliliters of semen per ejaculation. Average 120 million sperm cells per milliliter of semen. The male external reproductive organs include the scrotum and 2 testes and the penis. The scrotum is a pouch of skin and subcutaneous tissues. There's a dartos muscle in the subcutaneous tissue that contracts to cause wrinkling of the scrotum. There's a medial septum that divides the scrotum into 2 chambers. Each chamber is lined with a serous membrane. Each chamber houses the testes and epididymis. Okay, and then there's the penis itself, conveys urine and semen. Specialized become erect for insertion into the vagina. And then erection, orgasm, and ejaculation. Parasympathetic nerve impulses control erection. Blood accumulates in erectile tissues. The orgasm is culmination of sexual stimulation accompanied by emission and ejaculation. Emission is the movement of semen in the urethra. Ejaculation is the movement of semen out of the urethra. This is largely dependent on sympathetic nerve impulses. And here's a slide going through what happens during erection, orgasm, and ejaculation. And then here's the functions of the different male reproductive organs. And then a hormonal control of male reproductive functions. Hormones secreted by the hypothalamus, anterior pituitary gland, and testes control male reproductive functions. The hormones initiate and maintain sperm cell production and oversee development and maintenance of male sex characteristics. The hypothalamus controls maturation of sperm cells, development of male secondary sex characteristics like armpit hair. Negative feedback among hypothalamus, the anterior lobe of the pituitary gland, and the testes controls the concentration of testosterone. The male sex hormones are also called androgens. Interstitial cells in the testes produce most of these, but small amounts are made in the adrenal cortex, testosterone being the most important. Here's the actions of the testosterone: increased body hair, sometimes decreased growth of scalp hair, enlargement of the larynx and thickening of the vocal cords, thickening of the skin, increased muscular growth, and thickening and strengthening of the skeletal bones. And here's how regulation of the male sex hormones takes place. Androgens prevent the GnRH, gonadic reproductive hormone. It comes directly from the hypothalamus. That leads to a pathway, I guess, to follicle stimulating hormone and luteinizing hormone. The FSH stimulates meiosis in primary spermatocytes. The FSH stimulates secretion of inhibin by supporting cells. The luteinizing hormone, interstitial cells, stimulates interstitial cells to secrete androgens. And then the testes produce inhibin and testosterone, which are released in the bloodstream. And then androgens in the bloodstream stimulate the development of male secondary sex characteristics. The androgens prevent oversecretion of a GnRH and luteinizing hormone and follicle stimulating hormone. So let's talk quickly about the organs of the female reproductive system. And here's a slide showing the reproductive organs. And here's a slide of the ovaries. Several ligaments hold each ovary in position. The largest, called a broad ligament, is attached to the uterine tubes and uterus. The suspensory ligament holds the ovary at the upper end. The ovarian ligament is a rounded cord-like thickening of the broad ligament. Like the testes in the male fetus, the ovaries develop from the masses of tissue posterior to the parietal peritoneum near the developing kidney. They descend in locations just inferior to the pelvic brim, where they remain attached to the lateral pelvic wall. Tissue of an ovary can be divided into inner medulla and an outer cortex. The ovarian medulla is mostly composed of loose connective tissue and contains many blood vessels, lymphatic vessels, and nerve fibers. The ovarian cortex consists of more compact tissue and has a granular appearance due to tiny masses of cells called ovarian follicles. There's two types of, well, actually, this is prenatal. So during prenatal development of female, the oogonia, or oogonia, depending on how you want to pronounce it, divide by mitosis to produce more oogonia, and these develop in the primary oocytes, or egg cells. Each primary oocyte is closely surrounded by a layer of flattened epithelial cells called follicular cells, forming a primordial follicle. So early in development, the primary oocytes begin to undergo meiosis, but the process soon halts and does not continue until the individual reaches puberty. Once the primordial follicles appear, no new ones form. Instead, the number of oocytes in the ovary steadily declines as many of the oocytes degenerate. Of the several million oocytes that are formed in the embryo, only 1 million or so remain at time of birth, and only 400,000 present at puberty. Of these, probably fewer than 400 to 500 will be released from the ovary during the female's productive life, and probably fewer than 10 will go on to form a new individual. Oogenesis, that's a process of egg cell formation. You have a primary oocyte with 46 chromosomes. There's a first meiotic division, and then you have the secondary oocyte, 23 chromosomes, each with 2 chromatids. And then you have fertilization. The sperm cell attaches with its 23 chromosomes. There's a second meiotic division. And then you have a zygote. 46 chromosomes, 23 from the sperm, 23 from the egg. Each chromatid now an independent chromosome. Now, after that second meiotic division, the second polar body degenerates. In the first meiotic division, there's a secondary oocyte, but the first polar body either degenerates or is a second meiotic division with 2 polar body degenerating. Let's talk about follicle maturation. At puberty, the anterior pituitary gland secretes increased follicle-stimulating hormone, and the ovaries enlarge in response. With each reproductive cycle, some of the primordial follicles mature and become primary oocytes. As many as 20 primary follicles may begin maturing at any one time. One dominant follicle usually outgrows the others. Typically, only the dominant follicle fully develops and the others degenerate. Ovulation. As the follicle matures, its primary oocyte undergoes meiosis I, giving rise to a secondary oocyte in a first polar body. The process of ovulation releases these cells from the follicle. And here you go, a slide of what's going on during ovulation in a time sequence. And let's look at animation of the maturation of the follicle.
A primordial follicle consists of an oocyte surrounded by a single layer of squamous granulosa cells. A primordial follicle becomes a primary follicle as the granulosa cells become enlarged and cuboidal. The primary follicle enlarges, and the granulosa cells form more than one layer. The zona pellucida forms around the oocyte. The structure is called a secondary follicle when fluid-filled vesicles develop among the granulosa cells and a well-developed capsule, or theca, becomes apparent around the granulosa cells. The theca possesses internal and external layers. The structure is called a mature follicle when the fluid-filled vesicles form a single antrum. At full maturity, the oocyte is located in the cumulus mass. During ovulation, the oocyte is released from the follicle, along with some surrounding granulosa cells of the cumulus mass, called the corona radiata. Following ovulation, the granulosa cells divide rapidly and enlarge to form the yellow corpus luteum. When the corpus luteum degenerates, it forms the white corpus albicans.
Okay then. So here's a look at the internal accessory organs of the female. That includes the uterine tubes, the uterus, and the vagina. And here's a slide of the uterine tubes. You can see the secondary oocyte in the follicle, the ovarian ligament, the ovary itself, and the uterine tube leading to the body of the uterus. And here's the endometrium, where the oocyte, when it becomes a zygote, will be implanted. And here's the uterine tubes with their cilia, and the uterus itself, some slides of it. And here's a vagina in different parts. And female external reproductive organs: the labia majora, labia minora, clitoris, and vestibular glands. Labia majora and labia minora and the clitoris and the vestibule. And here's a slide of the different parts of erection, orgasm, and ejaculation. Arteries and erectile tissue dilate, vagina expands and elongates, and it follows the process. The parasympathetic nerve impulses from the sacral portion of the spinal cord. And here's a table of the functions of the female reproductive organs. From the ovary, produce oocytes and female sex hormones. The vestibular glands secrete fluid that moisten and lubricate the vestibule. So our hormonal control of female reproductive functions. Hormones are secreted by the hypothalamus, the anterior pituitary gland, and the ovaries that control the development and maintenance of female secondary sex characteristics, the maturation of female sex cells, and changes during the monthly reproductive cycle. And again, we're showing the GnRH from the hypothalamus to the pituitary, the follicle-stimulating hormone, the luteinizing hormone, the gonadotropins into the ovaries. Estrogens are released in the bloodstream. The estrogens inhibit oversecretion of gonadotropins. The GnRH in the bloodstream leads to the development of breasts. increased deposition of adipose tissue in breast, thighs, and buttocks, increased vascularization of the skin, stimulates the endometrium of the uterus to thicken. And here's a chart showing hormonal control of female secondary sex characteristics. You see progesterone secreted by the ovaries affects cyclical changes in the uterus and mammary glands. And here's the major events in a reproductive cycle. You should get to know these. Anterior pituitary gland secretes follicle-stimulating hormone and luteinizing hormone. The follicle FSH stimulates maturation of the follicle. The granulosa cells of the follicles produce and secrete estrogens. The estrogens maintain secondary sex traits and cause the endometrium to thicken. The anterior pituitary gland releases a surge of LH, which stimulates ovulation. The follicular and thecal cells become corpus luteum cells, which secrete estrogens and progesterone. Estrogens continue to stimulate uterine wall development. Progesterone stimulates the endometrium to become more glandular and vascular. The estrogen and progesterone inhibit secretion of FSH and LH. If the secondary oocyte is not fertilized, the corpus luteum degenerates and no longer secretes. And as the concentration of luteal hormones decline, blood vessels in the endometrium constrict, and the uterine lining disintegrates and sloughs off, producing a menstrual flow. And then the pituitary gland is no longer inhibited and again secretes FSH and LH, and the reproductive cycle repeats. And here's a slide of the number of days and the levels of estrogens and progesterone. And developing follicle all the way to the corpus albicans. Menopause, it usually occurs in late 40s and early 50s. Reproductive cycles stop. Ovaries no longer produce as much estrogens and progesterone as previously. Some female secondary sex characteristics may disappear. It may produce hot flashes and fatigue, migraine, headaches, backaches. Also, hormone therapy may prevent effects on bone tissue. The mammary glands are accessory organs of the female reproductive system, specialized to secrete milk following pregnancy. Here's a location of the glands. And the structure, 15 to 20 irregularly shaped lobes. Out there, alveolar glands, drained by alveolar ducts. Which drain into a lactoferous duct that leaves the nipple and opens to the outside. Dense strands of connective tissue form sensory ligaments that support the breast. The mammary glands of males and females are similar. As puberty reach, ovarian hormones stimulate development of glands in females. Birth control is just a voluntary regulation of the number of offspring produced in the time you're conceived. This control requires a method of contraception. And here's a list of different birth control methods, all the way from the condom to withdrawal to the intrauterine device. Gives the mechanism, advantages, disadvantages, the amount of pregnancies per year per 100 women. Coitus interruptus, that chart talked about that. Rhythm method, 2 days before and 1 day after ovulation. Mechanical barriers, chemical barriers, and here's just a slide of different types of contraceptives. Combined hormone contraceptives, delivers estrogen and progesterone to prevent pregnancy. Here's an injectable Depo-Provera, prevents pregnancy for 3 months by preventing maturation and release of a secondary oocyte. Intrauterine devices or IUDs interfere with the implantation of a blastocyst. And surgical methods, you can cut and ligate the ductus or the vas deferens, which prevents sperm cells from leaving the scrotum. Or you can cut and ligate uterine tubes, and what will happen there is either the egg can get interrupted or the passage of sperm can get interrupted. Let's talk about sexually transmitted disease, or STDs. Most are bacterial and can be cured. Herpes, warts, and AIDS can't, or they're viral. Any cause infertility, and here's some of the symptoms listed: burning sensation, discharge, pain, inflammation, sores, blisters, bumps, rashes, itchy, runny eyes. And here's some of the sexually transmitted infections. Chlamydia is one that very often goes undiagnosed. It can cause pelvic inflammatory disease, infertility, arthritis, and ectopic pregnancy. Then you have your genital herpes, genital warts, which is human papilloma virus, gonorrhea and syphilis, which are two bacterial infections. Okay, let's look at the outcomes. State the general function of the male and female reproductive systems, outline the process of meiosis, and explain how it mixes up parental genes. The organs of the male reproduction, describe the functions of each part of the male reproductive system, outline the process of spermatogenesis, describe semen production exit from the body, describe the structure of the penis, explain how its parts produce an erection. Hormonal control, explain how the hormones control the activities of the male reproductive organs and development of males' secondary sex characteristics. Then the organs of the female reproductive system, describe the functions of each part, outline the process of eugenesis, explain how our hormones control the activities of female reproductive organs and development of female secondary sex characteristics. Describe the major events that occurred during a reproductive cycle. The mammary glands, review the structure, the birth control, describe several methods of birth control, and list the general symptoms of sexually transmitted diseases. And that's it. I had to rush through it because I didn't have enough time for both. I tried to squeeze it in. I ran over. Sorry about that. And I have to take a look to see what we have, if anything, for next week. We'll take a look at that right now. Well, next week we may or may not have a okay, no, next week you don't have anything because you have a final exam, or I'm a week early. So I might review a little bit of different chapters next week. I don't know exactly what's going to happen, but I might do that. Might do a little, make a PowerPoint, different parts of different chapters. Okay, well, thanks for listening.